All right, so let's start. Hello, everyone. Thanks for joining episode 8 of Warsaw.ai. It's our second remote meeting, second webinar. And as you know, this webinar is dedicated to applications of AI in fight against COVID-19, SARS-CoV-2 virus. So our first speaker will be Farah Shamout from Abu Dhabi, United Arab Emirates. She will give a talk about predicting COVID-19 related adverse events using chest X-rays and deep learning. Later, Grzegorz Preibisch from COVID Genomics will tell you about AI-based prediction of SARS-CoV-2 mutations. And finally, Mikołaj Błaż from Molecule One will tell you about AI-powered synthesis planning against COVID-19. So each talk will last about 20 minutes, and after each talk there will be about 5 minutes for Q&A session. This meeting is recorded, so we'll also later prepare a video and publish it on our YouTube channel and send a link to all participants. And if you have any questions during the lectures, please use the chat to just post your questions. So our speakers, the lecturers, may also try to answer your questions during the lecture, or just after the lecture during the Q&A session. All right, so now I will give away to our first speaker, Farah. So Farah, first of all, thanks for joining, first for agreeing to give a talk during Warsaw.ai. It's a pleasure to have you here. So we are happy to have you here. We are keen to hear about your research, and if you can please also tell us a bit about your institute, New York University, Abu Dhabi, about your research group, and so, yeah, we are just curious what you do to fight COVID-19. The Zoom is yours.
Thank you so much for the introduction. Good morning, good afternoon, good evening, wherever you are. I'm personally based in Abu Dhabi. My name is Farah Shamout. I'm an assistant professor, emerging scholar at NYU Abu Dhabi, and my research focuses on using AI to solve real-world clinical problems. So I actually joined NYU Abu Dhabi in September 2019. And I established the clinical AI lab. So it's still quite small. We're in the process of hiring one postdoc, but hopefully it will grow with time. And NYU Abu Dhabi is a portal campus of NYU that opened in the UAE since 2010. And the main campuses of NYU are in New York, Abu Dhabi, and Shanghai. So if you're interested about learning more about NYU Abu Dhabi, feel free to reach out later. And what I'm presenting today is a project that I've been involved in over the past 4 months with a team spread across NYU Langone Health, which is our School of Medicine, the NYU Center for Data Science, and myself in Abu Dhabi. And the title of my presentation, as you can see on the slide, is an AI system for predicting the deterioration of patients with COVID-19 in the emergency department. So feel free to send questions during the presentation. I'll try to answer them. I'm not sure I can see now I can see the chat window. Yeah, so feel free to send them throughout, and I'll try to answer them during the presentation or after the talk. So before I get into the details, just to give you some context on why we are doing this. So as you all know, COVID-19 is now a global pandemic. There are approximately 13 million confirmed cases globally, including 600,000 deaths, according to the World Health Organization. And the figure that I'm showing is the number of cases in New York in particular. And we started working on this project in the late week of March. So it's approximately a few days or weeks before the spike occurred in New York City. So now I'm going to discuss the motivation of the project. So although the pandemic has affected all of our lives in different ways, it particularly significantly affected hospitals. So this is a typical patient trajectory. So as you can see, if a patient arrives to the ED with serious symptoms, such as shortness of breath, fever, or chest pain, which may require some hospitalization in order to stabilize the condition. The patient could get admitted into the hospital. So this way the patient would be assigned a bed, for example. And then if the symptoms develop even further and the patient becomes seriously ill, they might get transferred into the ICU. And then if the situation gets even worse and the patient's lungs collapse, they might get intubated. And this requires the insertion of a tube through their mouth into the airway, and then they would rely on mechanical ventilation. And then eventually, if respiratory dysfunction occurs and the situation gets even more severe, this leads to serious outcome like a morbidity or mortality. And this is just the trajectory of one patient. Now, the issue here is that with the increase and the spike in COVID-19 cases, this imposed additional pressure on hospital resources, which were already strained even prior to the pandemic. So this is exactly what motivated us to pursue this project. And we primarily decided to focus on the emergency department because in the emergency department, there's a lot of critical decision-making that takes place, especially for patients that need immediate care. And this then determines their future trajectory. So the main goal is when a patient arrives to the ED, what is the risk of deteriorating in the near future? And by near future here, we refer to 24 hours up to 96 hours. So within 1 to 4 days, what is the risk that this patient would deteriorate? Now, the next question is, how will this risk evolve over time? So we're not just interested in the overall risk score of the patient, but we're also interested in how the risk might evolve over time until the patient gets discharged. Based on our conversations and discussions with frontline workers who are actually involved in with fighting the pandemic and others in the hospital, we decided to focus on 3 main outcomes, which are ICU admission, intubation, and mortality. So we define deterioration as the occurrence of any of those 3 outcomes. Now, what makes healthcare so appealing to use digital technologies such as AI is the availability of data, and vast and different types of data. In the emergency department, for example, for example, vital signs could get collected, like the patient's heart rate, blood pressure, respiratory rate, etc. And actually this specific type of data, like vital signs and labs, have been extensively studied prior to COVID-19 as strong predictors of deterioration. So a lot of the existing risk prediction scores, which are often referred to as early warning score systems, do rely on those vital signs or lab tests. Chest x-rays is what we decided to focus on along with those other types of variables, because chest x-rays are considered as the first, they're used as a tool to triage patients once they arrive to the hospital based on the interpretation of the radiologist of the image. And on average, vital signs are collected prior to chest x-rays and labs. Labs are usually ordered when the patient arrives to the hospital, but then it takes some time until the results are out. So this is just to give you also, just to give you an impression of the sequence of events in terms of data collection. Now, why chest x-ray imaging? So as I mentioned, the other clinical variables have been studied extensively, and you'll see that there are a lot of papers that have been published in using those clinical variables for patient prognostics, so to predict deterioration. Chest x-rays in particular have not been used extensively, although they play a very important role in triaging patients, so deciding whether to admit a patient or not. The disease itself manifests as a respiratory illness, which means that chest x-rays could contain useful information. Compared to other imaging modalities like CT scans, which a lot of papers have also been published recently on the use of CT scans, chest x-rays are cheaper and easier to obtain, and portable chest x-rays do not require transferring the patient to another imaging suite. So they can just roll it in and take the chest x-ray image while the patient is on their bed. And so this does not risk decontamination, for example, like what CT scans may impose. So we have two main tasks, as I mentioned a few slides earlier. The first task is to predict the overall risk of deterioration, and we define deterioration by the three main outcomes that I mentioned. So given the patient's chest X-ray, what is the probability that the patient may deteriorate in the next 24, 48, 72, or 96 hours? And those are the ground truth labels that the patient, whether the patient had an adverse event or not. And so we predict the probability estimates as shown in the bottom of the slide. Now, the second task is to predict how this risk may deteriorate over time by producing deterioration risk curves. So those deterioration risk curves can then show how the patient's risk evolution compares to other patients, for example, who are currently being assessed in the ED. And we chose 8 time windows or time buckets to estimate the probability of deterioration within this context. Now, the data set was collected at NYU Langone Health, or the School of Medicine, and we originally had around 20,000 chest X-ray exams, but then since we're focusing on the ED, we applied a rigorous inclusion and exclusion criteria in order to get the final training and test sets. So we had around 2,900 patients in the training set and 718 patients in the test set, which had around 5,600 images in the training set and 830 images in the test set. So this is just to give you a sense of what the data looks like. And for each of those chest X-rays, we also extracted their clinical variables, which are the vital signs and the labs. So now, how do we learn from the chest X-rays? So we applied the globally aware multiple instance classifier, which was published by Shen et al. Shen or RT is also part of our team. And I'll give you just a quick overview of the architecture, but basically it consists of 3 main components. The first component is the global module. So given an image, some global network, which we parameterized here as ResNet 18, extracts some features, some representation, which then provides saliency maps. And we note here that we actually do not have ground truth localization labels. So this is also a big advantage. And this component of saliency maps is also one of the reasons why we went for this architecture, because we really wanted to incorporate interpretability within our system. And then the second module relies on regions of interest patches that are extracted from the saliency maps through a greedy algorithm, which then get further processed through another local network, which is also a ResNet 18. And then we assign attention weights to each representation of those regions of interest patches, which then get concatenated and processed in the final module, which we refer to as the fusion module. And then at inference time, we rely on this prediction, diffused prediction. Now, to give you a sense of the loss function, this is the loss function of the model, and it consists of 4 main components. So the first component compares Y global to the ground truth labels. The second component compares Y local to the ground truth labels, and the third component compares the fused prediction to the ground truth labels. And finally, we apply L1 regularization on the saliency maps. And so this is the overall loss function. For the survival curves or the deterioration risk curves, the loss function is slightly different, but I won't go into the details for brevity and for time reasons. And then, so that was for the chest X-rays. Now for the clinical variables, we decided to use a commonly used model for such variables, which is gradient boosting models. And the reason why we went for that is that based on my experience and based on other papers, they just perform best for this kind of data that might have its own issues, like missingness, for example. And it's also computationally efficient and offers some form of interpretability. So for example, here I'm showing just for the 24-hour window, the most predictive features of deterioration, temperature, heart rate, respiratory rate, age, etc. And those are actually aligned as well with clinical findings of what might predict deterioration for COVID-19 and other general patients. Now, how do we ensemble the predictions? So we actually do it in a simple way, where we just do a weighted sum of the chest X-ray prediction, this is the chest X-ray prediction and the clinical variables-based prediction, and we optimize lambda on the validation set for each time window. So now what are the results, which is the exciting part? So first, our results suggest that chest X-rays do actually contain useful information for patient prognostics. So on the 96-hour window, we achieve an AUC of 0.738 and the PR AUC of 0.439. And actually, based on our conversation with the clinicians, they also informed us that the 96 hours prediction is the most useful one for them, because for shorter time windows, they sort of already know or can tell that the patient is in a bad condition. So now, in order to offer some interpretability, we also assess the saliency maps that were extracted through the GMIC model. And in general, when we showed this to radiologists, we could tell that the saliency maps highlights regions of lung consolidation, or this is when the lungs are filled with, lung tissues filled with liquid instead of air. So it actually clinically makes sense. So as you can tell, it highlights some regions of the left and right lung. One thing to highlight here is, for example, in this case, or in the third case, the model doesn't highlight all of the regions that are associated with like deterioration or the severity of the disease. And this suggests that our model really focuses on particular regions that are most associated with it or informative of its prediction. However, the role of the radiologist is really important to get a holistic interpretation of the image. So in fact, they would actually cover, their interpretation would cover the entire lung space. As for the regions of interest, so looking at the first row as an example, this patch corresponds to this patch here, and this patch corresponds to the patch on the other side, on the other lung. And they get assigned the highest attention scores. So this also helps prioritize which regions within the saliency map are most associated with the deterioration, which is also useful for interpreting the model. Now, the second important finding is that although clinical variables have been mainly used for risk prediction, even prior to the pandemic, both chest X-rays and clinical variables contain complementary information. So the best performance was achieved when we combined both models through the ensemble that I mentioned earlier. So those are the deterioration risk curves. So the red line shows, so actually the faded red lines show the deterioration risk curves in the test set for any patient who experienced an adverse event. And the blue lines show the deterioration risk curves for anyone who did not experience an adverse event. And then the blue bold line is the average for all of that, and the red bold line is the average for those who experienced an event. As you can see, the model can effectively separate both of them. And the black line is the average deterioration risk curve for all patients in the test set. And we also assessed the model's performance in this context through the concordance index, and it achieves concordance index above 0.7. Another aspect of our study is that we actually performed a reader study. So we asked 2 radiologists to label around 200 images of whether they think they will experience deterioration in the next 24, 48, 72, or 96 hours. There are a few takeaways from this. First, regardless of experience, so we've got here 3 and 17, experience does not affect the radiologist's performance in this context, and this actually emphasizes the fact that the clinical understanding of the coronavirus has not been fully established yet. And then we also compared our model to the average of both, the predictions of both radiologists. Now, the average of the prediction of both radiologists outperforms either of them, which is expected. And because such annotations or interpretations of images by radiologists are subjective, so they both probably picked up on different parts of the image. And then our model, COVID-GMIC, even outperforms this average prediction of both radiologists. Now, to get to all of those results, we actually performed a lot of experiments. So we compared GMIC to other baseline models like DenseNet. We also compared gradient boosting models to logistic regression and random forests. We did some ablation studies where we looked at the size of the image, size of the training set. We did a lot of hyperparameter tuning. And finally, since our data set is quite small for an imaging-based application, we actually also tried pre-training the model first with a generic chest X-ray data set that I think diagnoses 14 diseases, and this improved the performance of the model through transfer learning. Now, the implications of our study. First, this suggests that chest X-rays do contain useful information for the risk prediction task. The interpretability component in particular has the potential to perhaps enable a better understanding of the disease, since from a clinical perspective that hasn't been fully established yet. We also actually tried deploying one of the simpler versions of our model within the hospital infrastructure, just to see whether we would face any logistical or organizational challenges in terms of deploying the model. And without using any GPUs, it actually takes 2 seconds to process one DICOM file, which is the format in which an image chest X-ray image is stored as, and to compute the model prediction. This can support our future work and invest in prospective validation studies. So to summarize, we use data from NYU Langone Health, both chest X-ray images and clinical variables, in order to predict deterioration. And we evaluated our model through retrospective evaluation on the held-up test set, through the reader study, and by deploying it silently in the hospital infrastructure. And this is what we offer the clinicians for assistance. I would like to just thank all of the team members, because we've been working on this extensively for the past 4 months. We have a great team of both computer scientists, engineers, and clinicians, radiologists, even medical students, and it's been a great experience overall. And the team, as I mentioned, is in collaboration between NYU Langone Health, the NYU Center for Data Science, and NYU Abu Dhabi. Thank you for listening, and I'm happy to take any questions now.
Okay, thank you very much, Farah. Very interesting lecture. Are there any questions? Because I have a few, but maybe someone else would like to ask as well. I see that we don't have any questions in our chat, so maybe I will ask the first question. You said that your model can be potentially easily deployed in a hospital, right? Do you already maybe collaborate with any hospital at the moment, or are you planning such collaboration? How do you assess how difficult it might be to, or how much time you may need to just apply your models in practice?
Mm-hmm. So actually, what I mentioned is we tried deploying it in the hospital. So
Okay.
this is on the hospital infrastructure of NYU. It was silently deployed, so the radiologists weren't really using it. We just wanted to see if it's feasible to deploy them. And the results I showed in terms of processing the image within 2 seconds is after we deployed that model silently without having any GPUs as well. So it was on a CPU-based computer.
Okay, perfect. You also mentioned that the model that was a combination of a neural network and gradient boosting model outperformed both models applied separately. And there was a linear combination, right? You said that you are just looking for the optimal value of the lambda parameter, right? Can you tell us what was the optimal, the best value for this lambda, for the proportion? Do you remember?
That's a good question. I don't fully remember them, but I can go back to the figure. So over here, here I'm showing the optimal lambda per time window.
Okay.
So for 96 hours, it's around 0.62, I think. And then for, let's say, 48 hours, it's a bit higher, like 0.8. So I don't think there's like a specific pattern here. It really depends on what each model has learned within those time windows.
Okay. Yeah, because I guess that both models have also some hyperparameters probably, right? So I guess that you probably also perform some hyperparameter tuning, right? Okay. Okay, but this description of both models can be found in the paper which you referenced at the end, right?
Yeah, and I would like to point out that we still haven't posted the paper. We're posting it in the next few days. We're just polishing last. Please keep an eye. It will be posted soon.
Okay, and where it will be published?
So we're going to post it on archive while it's under review in the journal. Okay,
great, fantastic. And by the way, I see that you collaborated with Krzysztof Geras and Stanisław Jastrzębski. They were our speakers during the past Warsaw.ai events. Actually, Krzysztof was a speaker during the previous Warsaw.ai meetup in March this year. So good to see them as co-authors as well. And our third speaker, Mikołaj, is also collaborating now with Stanisław Jastrzębski in another project. So maybe he'll also tell about it. Okay, are there any more questions to Farah?
Okay, I have two questions. First of all, can you hear me?
Yes, we can hear you.
Okay, so first of all, thank you for a great talk. And my first question is about the model itself and the training of the model. If I understand correctly, you use a combined loss when you add loss from the global model for the saliency map and the local model. So my question is it necessary to use this combined loss? Wouldn't it be enough just to minimize, just to maximize the probability of the correct class, and just use all the models combined to just minimize this single loss instead of also minimizing loss for saliency map? So that's the first question. And the reason I'm asking this is that I imagine that the model could then learn how to invent those maps itself, just to minimize the loss. Okay, so that's the first question. And the second question is, because you said you evaluate on a held out test set, so when you construct this test set, is it like randomly sampled, or do you account, for instance, on different, I don't know, hospitals or machines? Because I've read that these models learning on X-rays, they often have problems to generalize on new hospitals or new types of machines, for instance. So do you also like test how well you generalize to such specific cases? And yeah, and that's both questions. So thank you.
Thank you for your questions. So for the first question, I think we did run an experiment where we just had like one of the modules separately, but the model performed best when we had all of them. I think the paper I mentioned goes into details about how the loss function was defined.
Farah, sorry, Stan here. So I can actually give one more detail about this. I think it's an interesting deep learning detail in the GMIC model, if you don't mind. So the thing is that there are no gradients passing from the local model into the ways of the backbone of the network, because there is a non-differentiable operation inside. So that's the key reason that we are not backpropagating there, because it's impossible, as far as I understand your question.
Yes, okay, I get it.
Yes. So it's actually an interesting thing just from the practitioner deep learning perspective, in the sense that this model does something weird where you see the retrieve ROI operator. It's non-differentiable because it's taking like top 5 patches from the model, which is a small Python function to do that. And that's something that the main author of this paper, I think, is looking at, trying to avoid and backpropagate for everything. So just to complement Farah's answer.
Thank you, Stan. For the second question about the held out test set, we split it based on patients. So we had the chest x-rays coming from the same patient were either only in the training set or the test set. We did not really do any performance assessment across different hospitals, let's say, or different machines, but that's definitely an interesting point of doing that in future work, because as you mentioned, it might not generalize well across different machines due to, let's say, the resolution or the patient cohort or the characteristics or the demographics of the patient population. But it's definitely, I think, a really good direction for future work to see how those models may generalize in a different patient cohort.
Okay. Thank you.
Thank you very much. All right. Are there any more questions? If not, then I think that we can start our second talk. So thank you, Farah, very much once again. I hope that you will stay with us, so maybe some other, so people can maybe also later ask some questions on chat. But now let's give a voice and zoom to Grzegorz Preibisch, our second speaker from COVID Genomics, Polish startup from Warsaw.
Hello.
Yes, we can hear you.
Okay, I will try to share a screen. Can you see a screen?
Yes, we can see it. Perfect.
Okay. Okay, so in the beginning, I would like to introduce myself. My name is Grzegorz Preibisch, and I'm a medical student at the Medical University of Warsaw, and I just finished 4th year, and also I'm studying mathematics at University of Warsaw, and I just finished 3rd year. During this year and during the pandemic, together with Peter Grzegorczyk, we developed a startup which focuses on analyzing SARS-CoV-2 mutations and evolution of that virus. And well, we have won some hackathons. We had 1st place and Hack the Crisis, and 3rd place in EU versus virus. Also, during the speech, I will try to talk about the model which we developed and the Stanford Researchathon, and also a little bit of our work after the researchathon. Some work isn't finished yet, so I will show you the general view of the model. And also, I'd like to say that I'm much more oriented in medicine and stochastic processes than in machine learning, but let's talk about the model. At first, I have to define the problem. We wanted to focus on finding the best vaccine candidate for COVID-19. But the problem is that most papers focused on analyzing that data, which is basically too small to conclude anything about evolution of virus. In some papers, they took 5 to 20 genomes, and that approach may be irrelevant and may not work, for example, if a virus will evolve. In our model, we tried to focus in a new type of vaccines, which are mRNA vaccines, and they need a little bit different approach than the traditional one. So basically, what we have done, we built up a pipeline which is learning on much bigger data set than the most people did. For comparison, two papers which I referred to, they analyzed 40 of genomes. We analyzed 40,000 of genomes. So I'll show you the general idea. What kind of process stays behind the how this vaccine might work. Basically, we have to explain a little bit about biology. The mRNA vaccine is generally working by learning immune system using to use cellular response, which is basically the immune T-cell response, and it needs. I will describe you briefly how it works. So basically, in the beginning, we have healthy cells, and they get infected by the virus, and then they try to reproduce the virus, and to do that, they need to produce viral proteins. Our cells try to fight with the virus, and that's why they cleavage the proteins and start to present them on the HLA receptors. And if they present them on the HLA receptors, immune T cells can kill the infected cells and stop the infection. So that's why we built up a pipeline to manage this problem. But in the beginning, I would like to say something a little bit about the data. We took genomes from GZ repository, which contains genomic data, which we can briefly explain as strings in five-letter alphabet in FASTA format with date and place where a sample was collected. And also we can translate that into proteomic data by simply applying a function, which is really simple. But first, before using that data, we have to filter that data. And that was a big problem because a lot of laboratories which put genomic data haven't put good quality data. They put data with a lot of errors, and we have to clean that up before making the model. So proper region of genome should contain only 4 letters, and N means that's an error, or we don't know what is it. And that's a technical problem. So basically, briefly speaking, we developed in previous projects a pipeline which filters data by defining the region of interest, for example, gene or subregion of gene. And we find that region using heuristics and count N and decide then what, and reshape the model for the purpose of our model. In that case, we excluded all possible data which contains any N in the spike protein and analyzed structural proteins which may be useful in immune response. Our pipeline is made on is using 2 machine learning tools already developed in recent years. One is NetChop, which at first cuts proteins from the genome set, which I described the way of creating previously, and then the output from NetChop we have put that in a NetMHC-PAN, and from that we made a big data set. And the most important thing is that we're trying to optimize the probability of existence of peptide, because in some way peptide maybe exist or not with some probability, and that's an important fact. And also the second fact is that maybe that peptide may be presented with good or bad measure, I would say. So on this plot, we can see that if we take into consideration two HLA types, which vary a lot across the populations, those two are the most popular ones in the UK. If we look at them, they can really have different binding level across all possible genomes on only describing one peptide. So the problem is that we have to find peptide which is not mutating, and also a peptide which has good presentation on each of HLI which we choose to in a different country, because it differences across the ethnicity and geographical region. That's why we have to choose HLA types to have a good vaccine by choosing HLA based on the country. And that's why we have chosen to some score which we can briefly explain by multiplying probabilities of existence of peptide, probability of presentation of one peptide on HLA, and also normalized mutation rate. As I previously described, existence of probability of existence and presentation of that peptide on HLA are outsourced by other tools, but normalized mutation rate is computed by taking a parameter of Poisson process from the genomic data. That's why we made some big data set with labels described with each peptide which was possible to make from the data set, and also that one score, and that's what then we used XGBoost. So the features as I described are peptide in that data set, peptide, which is categorical, position in protein, that's an integer. That's the integer which represents the beginning of peptide. Edit distance, which is the some way of measure of mutation rate, and also HLA type, region, and date. And a label was mutation aware binding score, which I just previously explained. So train set was collected before 1st of April, and samples were collected after 1st of April, and we did that in the beginning of June. So we trained that set on 30 millions of possible peptides and obtained an MRC of 0.034, and performed a chi-squared test to reject the null hypothesis, and model performed quite well, but I wasn't sure if that's all what we can do to predict, make some good measure of peptides. So that's why we started to make next pipeline. Basically, the pipeline is a little bit different because we don't use XGBoost. We just try to use LSTM to predict evolution of proteins. And the inspiration for that was a paper from the January of this year. In that paper, they described the new neural network, which was much better than traditional methods to predict influenza virus. And that's why we at COVID Genomics decided to build up LSTM. And unfortunately, the model wasn't really well working, because the result was that most of places won't be changed, won't get changed, because the data which we fed into LSTM was basically taking a sample with date and positions which changed. And a lot of proteins, most of them haven't changed. That's quite of not surprising result that the model haven't performed well. But the general problem why it haven't performed well is because and performed well in that paper is that basically SARS-CoV-2 virus mutates and at lower rate than influenza. And the second problem is that we have data from 20 years of influenza. That's why model haven't performed really well, but that haven't really discouraged me to make that model further, and that's why we came up with next approaches. The next approaches which we are going to do are using GANs to build distribution of location of mutations, then use that distribution to build stochastic model. From my experience, stochastic models are much better in the when data isn't big enough to make machine learning. That's why I came up with that idea. And the second one is use much bigger data set and change our hypothesis. And from the LSTM model, I have some idea that the model wasn't so bad. We just learned that, teach that model in that way, I think. And what was overfitted was our thinking more than the model. And that's why during last week I came up with reading a lot of papers, and I came up with hypothesis and some idea to make a bigger data set. Basically, I would describe that it doesn't matter what virus is mutating, which virus is mutating, it escapes from immunological system, I would say. So I defined a measure which goes from one space, which describes of space of possible epitopes, which is the region which is attacked by immune system. And the measure says the conditional probability, which is regular, by the way, that B is not resistant to immune system, and under condition that is resistant to A. And the hypothesis is that the evolution of a virus is based on a conditional distribution of that measure. And the most probable strain is argmax of that measure. Also, I would say that there were 2 or 3 papers during last 2 or 3 years about that. And that's a general hard problem to solve, which I'm going to do with COVID genomics. And that's basically all what I have to say about the models which we developed. Also, I would like to say a little bit about our team. and say thanks to all people which helped in a Stanford hackathon. And also I would like to say special thanks to Igor Adamski, which helped with developing LSTM, and also Ivo John, who helped with developing LSTM. And of course, I would like to say thanks to Paweł Gorat for advising us a lot, and also to Professor Michalewski, which did the same. And from our discussion, I came up with a lot of ideas, and I just wanted to say thanks.
All right, and we also thank you, Grzegorz, for your lecture. Are there any questions to Grzegorz? I also don't see questions here on our chat. So maybe I'll ask first. So I understand that currently we are, unfortunately, we are not able to predict mutations of this SARS-CoV-2 virus, at least as accurately as in case of the influenza viruses, right?
Yeah, I mean, I would say that yeah, but that's good because of COVID isn't mutating as fast as influenza.
Mm-hmm.
And that's why we have much less data, because we have only half year, and virus isn't mutating so fast. That LSTM model may be useful in interpreting genomic, not immunological data, but in some manner that's good that virus isn't mutating that fast that probably it will prevent making vaccine, but it's hard to say. The problem is that that data is too flat to make machine learning. I think that making stochastic model would give some answer,
Mm-hmm.
but the problem is that even if one new strain will exist, we don't have an interpretation of that strain, if that strain will be resistant to vaccine or not. The measure which I described here will try to answer will give the meaning of mutation, and will try to describe what's the basically difference and define if that difference is enough to make resistant to vaccine.
Mm-hmm. All right, and can you maybe also explain why predicting the mutations is so important from a perspective of developing a successful vaccine or test for COVID?
Okay, so imagine that if we have one strain, and for example, one strain mutates, and we have vaccine against one strain. Basically, I would describe that using that measure. If we have strain A, and we have vaccine against strain A, there is maybe that's possible that strain B will be resistant to vaccine, and it will make a pandemic again, because basically, as we all know, SARS-CoV-2 virus spreads quite heavily. Mm-hmm. And existence of one resistant virus would mean the beginning again of pandemic.
Okay, so
I'm not sure if I answered well.
For me it's fine. So I understand that that's also one of the reasons why we need to develop, or we have to develop a new vaccine for influenza each year, right? Because as you said, influenza also mutates even faster than SARS-CoV-2, right? And how do you think, can we maybe already assess how good might be the vaccine once we develop it? So, for example, do you think that we'll have to develop and improve our vaccine each year, similarly as in case of influenza, or the vaccine may give a resistance for more than 1 year?
I think that the difference between SARS-CoV-2 and influenza would be I would say that the period of time which SARS-CoV-2 needs to mutate and get resistant will be a little bit longer. So we won't have a SARS-CoV-2 virus each year, but I think that's possible that we'll have a SARS-CoV-2 virus, new SARS-CoV-2 virus each, for example, 4 to 5 years, which I believe would be really bad. That's why we decided to tackle this problem.
Okay. And do you think, based on the current results, that it might be possible to develop a vaccine that may give a total resistance forever, for the whole life?
No, for a whole life, yeah. The problem is that there will be resistant for a whole life for one strain of SARS-CoV-2 virus, but we won't be resistant to all possible strains in the future, which maybe one will be resistant to our vaccine and escape from our immune system. That's why I think that there is a high probability that we will have to vaccinate, for example, each period of time. We have to say something that that's this third outbreak of coronavirus in this century. We had SARS-CoV-1, MERS, and that's the third one. So basically, maybe it will be a need of vaccine each 3 to 4 years, in my opinion.
All right. Okay. Are there any more questions to Grzegorz? If not, then I also thank you, Grzegorz, once again. It's really impressive what you are doing, especially that you are still a student and even bachelor student. So good luck in your research. And now let's welcome our third speaker, Mikołaj Błaż from Molecule One.
Hello, let me share my screen.
Okay. Okay, we can see your screen. You can start.
Cool. So hello, my name is Mikołaj Błaż, and today I'll talk about AI-powered synthesis planning against COVID-19. Thank you for an invitation for this talk. I work as a machine learning engineer at Molecule One. We are a startup which helps chemists design synthetic pathways. That's why this talk will be a bit chemistry oriented. What you see right now is the agenda. And I mean by that I will walk you through different subsets of the title. So let's start with singletons. What is AI? Okay, just kidding. I know I don't have to explain this one, but I might need to explain what synthesis planning is. So in a second, I will show you how to build a synthetic pathway for the target compound present on the screen. So a compound can be a potential drug, because a drug is simply a compound, a molecule. So just to be on the same page when it comes to nomenclature, this is a reaction that reacts from substrates to a product. And in order to build a synthesis plan, we have to gradually break the target compound into simpler ones until we reach compounds that are commercially available. And just to avoid confusion, I just show you how to build a synthesis plan in the so-called retro direction. So from the root to the leaves. And this is how you think about it in the synthesis planning task. But in the real world, so in the forward direction, you go to the shop and buy the substrate leaves, and then in the chemical lab, you gradually synthesize them to the intermediate products and then to the final product. So this might have been confusing. And another source of confusion is that a synthetic pathway is actually a tree of compounds. So you have been warned. Okay, so let's move to another singleton, COVID-19. You unfortunately know this guy, but I want to tell you about 3 different ways of how to tackle the SARS-CoV virus. So first one is drug repurposing, and by the way, this is the quickest one, where you take existing drugs and check if by accident they work against the virus. The second one is vaccine research, so it's connected to the topic of Grzegorz talk. And the third alternative is to design a drug from scratch. And this is the category we fall into simply because that's what we do. We design synthesis, we help design synthetic pathways for new drugs. Okay, so let's say you want to design a drug against COVID. What can synthesis planning do for you? So the first thing you need to know is that on average, drug development takes 12 years and costs $2 billion. So what you typically do is you scan multiple initial compounds, and then you have to actually physically synthesize typically 20,000 of them, and then you take them to the trials, and hopefully you end up with a single drug if you're successful. So I think it's not that surprising that with a good synthesis planning, you can drastically reduce both the costs and the time needed to develop a drug. And actually, synthesis plan becomes even more important in time, because just like we have Moore's law in computer science, we have inverse of it. It's Arum's law, and it's inverse both when it comes to spelling and meaning. So to paraphrase it, the cost of a new drug doubles every 9 years. So in the graph, you can see that the logarithmic number of new drugs decreases in time, actually. And I don't want to go into details about the reasons behind this, but I want to tell you that it's getting even worse, because people start to use deep learning to generate candidate compounds, so candidates for drugs. And it's a known fact that deep learning models propose hard to synthesize molecules. So you can easily end up with a synthesis plan that looks like this. And believe me, you don't want to take this plan and give it to your chemist and tell him to synthesize it. Okay, so let's go to the reason why we are all here: AI against COVID-19. So as an example, I will show you AI-powered. Oh wait, sorry, bad direction. Don't try this at home. Next slide, please. Okay, I meant AI-powered synthesis plan. And now it's becoming interesting. And by the way, if you say AI-powered synthesis planning, you can as well say molecule 1, because essentially that's what we do. So I would like to start with a statement that synthesis planning is the goal of organic chemistry, except that it's harder. Let me explain it. So our goal is to find a synthesis plan for a given compound. And sometimes we want it to be possibly cheap, but not always. So we might represent this problem as a tree search problem. So we have to search a tree where for each compound, we have multiple alternative reactions. So we have multiple moves in this game. And we have to choose the reactions that break the target compound into the biopurchasable substrate leaves. And we usually look for solutions of maximum depth of 5 or 6. So you're probably thinking now, it can be right. This problem doesn't sound that hard, right? Because even if the branching factor, so the number of possible moves at each node, even if the branching factor was like 100, if we are looking for such shallow solutions, this problem sounds feasible. And partially, you're correct. You can employ the pretty standard search algorithm like A*, which, by the way, we use. People also use MCTS or proof number search, and you obtain good results in this problem of tree search. So where's the catch, right? So the biggest challenge here is that actually we don't know the correct moves. So the reactions at each node that are used to break compounds. Why is that? So imagine that in a synthesis plan, we use this reaction. So from the substrate at the top to the rightmost product. And then you give this plan to your chemist, who returns you after like 2 days and says that those substrates didn't react at all. Or maybe those substrates gave a different product, different one. For example, the leftmost one. And this is a problem because we don't even have a simulator to validate the reactions. So to validate each reactions, we have to spend a lot of resources. So the only thing we have to do is rely on the data that we already have about the already carried out reactions. But the bad news is that we don't have much. We don't have much data. It's the order of millions of reactions, whereas AlphaGo needed billions of moves to train properly. So people try to solve this problem in different ways, and we have our own solution too. Recently, we developed and published Megan. It's a neural generator that generates candidate reactions. So essentially, this is the moves generator for this game. And it works the following. You input the target molecule, the one that you want to break. We embed it because we are operating on graphs, encode it once, and then with a stateful decoder, we decode a sequence of actions that are needed to be taken to break this molecule. And each action is a small edit to this graph, like adding a bond or removing an atom. So in this example, the decoder says that the first action is deleting this bond. And this is actually the step in which we break the product into two separate substrates. Then the next action is editing this nitrogen atom. This is a technicality, but still it's needing for the substance to be valid, but still the model has to do it explicitly. Then we add iodine atom to the other substrate, and then the decoder says to stop. And this is how we obtain the first prediction for this compound. So the first move. And with beam search, we can generate as many moves as we want. Typically, it would be 10 to 100 of moves. So this model is template free. I don't want to go into details what it means, but in the context of COVID, it's important to know that it can propose uncommon reactions, which is particularly useful when you are operating on novel chemistry. This model was trained with imitation learning on the open access dataset, so the dataset of reactions extracted from chemical patents. You might be interested how to evaluate models in this task. So let's say we have a data set of ground truth reactions, and we can produce two tasks from it. The first one is guessing the substrates given a product, and it's a single-step retrosynthesis prediction because we we don't predict the whole synthesis path, we just predict a single step. And the second task is predicting a product from given substrates. So if those two substrates react, what will be the product of this reaction? And this is forward synthesis prediction. And in both tasks, we use top K accuracy as a metric. So yeah. And when it comes to benchmarks, I want to focus on 2 points. So first one is that with Megan, we are able to achieve state-of-the-art results for large K values on both tasks. What it means, we might not be the best at guessing the top move in retrosynthesis task, but when examining large number of moves, let's say 10, we are able to cover large part of reaction space. And the second point here, and by the way, the daggers mark concurrent work. So these are different papers. 2 of them were published very recently, including the purple line. Returning to my second point, those tasks, the retrosynthesis and forward synthesis prediction, look very similar, but they are actually very different. So forward synthesis is trying to predict what will be the result of reaction in real world, right? So it's the real world prediction, we can say. And retrosynthesis involves some strategy, how to break the compound efficiently. So we can observe that those 2 tasks are actually very different by observing the fact that no architectures present in the left-hand side graph are also good in the forward synthesis task, except from our model, which is good, which performs well in both tasks. But it reveals that modeling in chemistry is pretty hard, because it requires both deep learning knowledge, but also incorporating domain expertise into the model architecture as a prior. So think of convolutions in computer vision tasks. They drastically help models operate well on images, and here we have similar situations. All those model architectures, except maybe from the trans one, which I will talk about in a second, but all those architectures involve explicitly or implicitly some chemistry prior, like predicting reaction center, so the place where the reaction actually occurs. The exception is, as I said, the trans model or the molecular transformer model on the right. They're both not graph-based. They are text-based. They're actually translation models used by Google, for example. So based on transformer architecture, and they operate on smiles. So a canonical text representation of chemical compounds. And it's interesting to see that it does work in this task, but does not achieve state-of-the-art performance, especially in the retrosynthesis task, the harder one, because in the simpler one it's pretty good, but still it makes some trivial mistakes because of the representation, the textual representation. Okay. Enough theory. Let's move to the final topic, the complete title. So AI-powered synthesis planning against COVID-19. Let's go back to the graph representing the drug development pipeline, and I want to focus now on the first stage, virtual screening. So in this step, we screen those compounds in search for the ones that will likely be able to bind to the virus. So we perform multiple physical simulations and then select the compounds that are likely to be successful as a drug. So the question is, can we use synthesis planning as we use it in later phases also in this phase? Because then, if we choose those 20,000 compounds, they will already be easy to synthesize. So we will not waste resources to try to synthesize them if they're difficult to synthesize. So the answer is yes, and this is something we are working on recently. It's the synthetic accessibility score. So it works by, for each compound, designing a synthetic pathway and mapping this pathway to a number. So this number tries to reflect the estimated synthesis costs in dollars. So it's a clue if the target compound is easy to synthesize or hard to synthesize. And with this score, we are able to screen 5,000 compounds per hour. So as you can see, now we can bring synthesis planning also to the phase where we need to examine a very large number of compounds. So how it relates to COVID? Well, coincidentally, we developed this feature in February 20. So 1 month before the start of pandemic, and we decided to open the API for COVID researchers. So anyone who works on developing a drug against COVID is able to use this API for free. You can check it out on this website. And by the way, it's still this program is still open. And up to date, we have screened over 100,000 of compounds with 5 significant researchers. And here on this histogram, you can see an example of running a batch of COVID-19 targets against our score. And if you recall that from this large number of initial molecules, we have to select like 1% of them. This histogram, so this run against the score, tells you that you can easily forget about the 80% of hard to synthesize molecules, because the score of 10 means that our software couldn't synthesize it. So you can reduce the number of the compounds that you look at by the factor of 5 with almost no extra cost. And one particular collaboration that I want to talk about is with Mila in the context of their Lambda 0 project. So Mila is a research group led by, among others, Joshua Benjo, and they aim to be able to screen much larger number of compounds here. So currently they are able to screen 10 to the power of 10, but they aim for much more. And they use MCDS to navigate the space of molecules, which is super large, by the way. This is only a fraction of it because it's estimated to be 10 to the power of 60. So what I can share with you about this collaboration is a concrete example of 2 compounds that, when it comes to their physical properties of binding with the virus, they are comparable. So this is the first one. It says that when it comes to the properties as a drug, they are both likely to work. But if you look at their synthesizability, the first one is not synthesized at all. So you have hard time synthesizing it at the lab if it's possible at all. Whereas the second one has a straightforward 3-step synthesis. You can see it right here. So it involves only 3 reactions, pretty simple substrates. So by looking at the synthesizability, you can choose the compounds that are as likely to be drugs, but also are easy to synthesize. Okay, the last combination is the empty subset. So it means there's nothing more to add. So maybe let's see what we have seen so far. So first of all, synthesis planning is needed thousands of times during drug discovery process, and we've seen that it's a hard problem. Secondly, pure AI is not enough. We need both deep learning expertise and chemistry expertise in order to tackle this problem successfully. And finally, we've seen concrete examples of how automated synthesis planning can significantly speed up drug research against COVID. And it's all possible thanks to our great team presented here. Thank you.
Okay, thank you very much, Mikołaj. Are there any questions to Mikołaj? Do we still have Stanisław with us? Yes, Staszek is also here. So I have one question, or in fact, I have two questions. The first is related to the Megan model, which you described. So you said that this is a graph model. So can you explain a bit more how the graph structure is used to build the architecture of the network? And the graph is, I understand the graph is somehow related to the structure of the compounds, probably, right?
Yes. So we, by the way, the architecture is
okay.
described in very detailedly in each work. So yes, we input the graph, which is a compound, the target compound, and the main workhorse here are graph convolutional layers. So you can think about it as the equivalent of convolutional layers. So they look at the at each step, so at each layer, they look at the neighborhood of each node, so of each atom in this graph, and gradually, sorry, I can't pronounce it. Hierarchically, they collect the, so in the encoder, for example, which involves those layers, they hierarchically collect the knowledge about the compound, and then decoder also uses those layers to predict the actions.
And how many layers do you need in practice in total? How deep are such models?
If I recall correctly, this is a few, like I think it's 16 for the retrosynthesis task, which is harder than the forward synthesis task, where we need like only a couple of them, like 8 or something like that.
Okay. And what's the typical size of the input? So, for example, size of the graph, the number of atoms.
So the number of atoms varies between, I don't know, 20 to a maximum of 100. And the graph is represented as a matrix.
Okay.
So
just yes, the matrix of neighbors, I guess, right?
Yeah, encoding the connectivity and also some properties of bonds, because bonds and atoms, because as you can see, some bonds are the single bonds. Some of them can be double or even triple bonds. And they have also some other chemical properties. So all of this is encoded in this input matrix.
Okay. And do you use any special ways of training such models, or and how much time do you need to for both tasks which you mentioned?
So the retrosynthesis task is harder, as I already said. It requires like maybe 30 hours of training on a single Tesla GPU on a data set of 1 million reactions. And the training is, yeah, we imitate, so for each reactions in the data set, we imitate the actions that are needed to take to obtain the ground truth substrate. So we have a deterministic procedure of generating those actions, and we perform a gradient-based training to recover this identical path of actions, because to recover the substrates from a product, you could reach those substrates in different ways. So in different order of actions. For example, here we could first add the iodine atom and then delete the bond, right? So we make sure that in the training set we have a deterministic order of those actions, and then it's a simple gradient-based learning. We try to avoid reinforcement learning because of the lack of data.
All right, thank you.
I have a question.
Yeah, go on.
At first, what is the format of molecules data? Maybe I missed that, but I'm not sure if that was said.
So if you manipulate molecules for data analysis or for storing, etc., then you use usually their canonical text representation, smiles. So you just, yeah, it's a sequence of letters and some special characters that represent the compound graph. But for example, for a transformer NLP model, that's enough. You just the task is to map the text smiles to another text smiles. So this is how you represent a compound. In all other architectures that I presented, including Megan, we use graph-based model. So it means we have to reconstruct a graph of compound from this text smiles, because when we extract those reactions from chemical patents, we also extract the text representation. So in order to be able to input the compound to the model, we have to convert the text representation to the graph representation, which is done by some third-party libraries in Python, RDKit. And then we transform this graph of a compound to this matrix representation. We do it by ourselves because we have some properties specific to the model. Does this answer your question?
Yeah. Thank you very much. Really nice presentation, by the way.
Thank you.
Okay, I see that there is a question from Jakub. Do you plan to use computational chemistry software like Gaussian to verify the molecules that are created instead of doing it in the wet lab?
Good question, thank you. So yeah, so I might have said that the only possibility of verifying a reaction is taking it to the lab. If you are an experienced organic chemist, you can probably figure out if the reaction is possible or not by your experience, because you've seen some similar reactions. But it's hard even for senior chemists. When it comes to computational methods of verification, they are not. It's not possible for them to be 100% correct. I think we have plans to incorporate such computations to the training process in order to further increase the performance of our model by using multiple tasks learning. But currently we don't use such methods.
All right. Any more questions to Mikołaj? Last chance. All right. We don't have more questions, so thank you, Mikołaj, once again. A really great talk. Yeah, that was our last talk today. I will just show some closing slides, but I also see that Grzegorz would like to announce something. Grzegorz, our second speaker, right?
Yes, I just wanted to say that we are looking for people to collaborate, and we are looking for whole teams to collaborate in meaning of making some spin-off project. We may help you with developing pipelines to analyze genomic data. And also we are looking for people which would like to make some interesting machine learning projects in our team. Basically, we have developed one tool which will be published tomorrow or this weekend, which helps to say if tests for COVID-19 are affected by evolution of virus, and which allows labs to verify tests. And we will have a lot of interesting other spin-off of that project. So if you're interested, DM me, and I would love to talk with you if you would like to do something with us. And thank you for your attention. And
All right, thank you, Grzegorz. Yeah, we have to finish. Thanks everyone once again for your attention. If you liked our webinar, if you like our events, let's stay in touch. Please follow our fan page, our Facebook group. You can join our mailing list and send us feedback. Most likely I will also send you a survey after this meeting to ask about your opinion and recommendations for the future. We are not sure yet about the next meeting. Most likely it will be in October. It should be October here, 2020, but we haven't decided yet, so we'll let you know. We'll announce it on our Facebook group and our mailing list. So thank you once again for your attention. Right now I'm stop sharing my screen, and I see that there's just one more question from Martin. How many chemistry people are learning AI? I'm not sure if any of our speakers can answer, or anyone else. I don't know personally how many chemistry people are learning AI. I think that I personally know one more person. He's not a participant of this meeting, but I know that he's doing PhD in chemistry as well, and is learning AI. So there are such people, even in Poland, even in Warsaw, but I don't know what might be the percentage of our chemists. Okay. All right, so now I'm also stopping recording.